So I'm actually excited to talk to this group. We've been working on methods for designing proteins for quite a number of years, and I think we're finally getting to the point where we can actually start to think about designing things that might be useful in the clinic. So my title here is Design of Protein Therapeutics, but that's somewhat fanciful. I think there are more immediate applications of protein design to diagnostics, sort of cheaper ways of making diagnostic devices, targeting, as well as therapeutics, and also be talking about materials. So what I'd like you all to think about as I'm talking is, what would be the best applications of the methods I'm going to tell you about? Because really, what we think about mainly are the methods, but what the best possible applications are of these methods to biomedicine. Probably many of you have a much better idea than I do. So we're always looking for collaborations and good problems to try and apply these methods to. In fact, the UW is going to be starting up an institute for protein design to really try and capitalize on the methods I'm going to tell you about to really bring these things into the clinic. So again, we need to know what your ideas for good problems and that where you could really make an impact on health. So I'm going to start by just describing sort of schematically the methods that we use for designing proteins, and then I'll go through sort of some of the specific applications. So the basic concept behind designing proteins is that we understand quite a bit about the physical chemistry of the interactions within and between proteins. So for example, we know that atoms in proteins have to be closely packed, that they can't overlap. We know a lot about the hydrogen bond. We know about the hydrophobic effect. And essentially, we've tried to encode our knowledge of these sort of fundamental physical forces in a program we've been developing over the years called Rosetta. And so we can calculate the energy of a particular configuration of the protein and or the energy of a particular interaction between proteins, and then change the sequence and try and make the energy lower, which would correspond to more favorable binding or more stable protein. So this is, roughly speaking, how we calculate the energy, just with equations that formalize what's shown here. And then we need ways now of varying the sequence. So the simplest possible case is, given a particular structure of a protein, for example, shown here in the ribbon diagram, we want to search over all possible sequences for this protein for the one that has the lowest energy as computed by summing up the hydrogen bonding, van der Waals interactions, and so forth. So this just shows you a movie of the actual computer calculation, a Rosetta calculation, searching over the different possible sequences for this protein. You'll see that most of the time when an amino acid is substituted in, the actual identity of the amino acid is changing too. So it's essentially just a random search for the lowest energy sequence. Now, in some applications, we don't really know what the backbone structure should be, so we're trying to search over backbone structures too. And in that case, we do it in two steps. We start by searching very broadly, just in a very approximate way, trying to cover the space that the protein the structure could sample, and then to calculate energies accurately, we need to put in all the atomic detail. And when we do that, we have to represent all the atoms in the protein. The calculations get very slow, so we typically do this in the end game. We have to do this, though, to be able to calculate energies accurately. So that's basically the sort of the schematic of the design process. Now I want to talk about the general process of designing molecular recognition. And again, perhaps the simplest thing to think about would be diagnostics. Suppose there's something a pathogen you want to be able to detect, or some other marker, and you want a protein that binds that target very specifically and with very high affinity, that you can make much more cheaply than you can make antibodies, and so you could use that as a part of a diagnostics device. So what do we do? Well, we first need to have the structure of the target protein. So if you want to detect flu, you need the structure of the flu protein. If you want to detect TB, you need something on the surface of TB. If it's some cancer biomarker you're trying to detect, then you need a structure of the cancer biomarker. And so either you have to have the structure, we have to be able to build a reasonable model of the structure. The next step is to design proteins which are predicted to bind with high affinity to the target. I'm going to be telling you more about how we do this soon, but conceptually, you sort of probably should already have the idea that we're going to have the structure of the target protein, and then we're going to try and make interactions with it, which are very low in energy using that force field I described. And really the key part, this is purely computational up to this point, but computation is cheap and not worth very much if you don't actually test it. So the next step is to, we build, actually just online from oligosynthesis companies, oligos to put together synthetic genes encoding these computationally designed proteins, or we just buy synthetic genes directly. And we typically, for any given problem, we typically make or purchase on the order of 50 different designs because the calculations we do are very far from perfect. Our model isn't perfect, so many of the designs that we make are not going to function as we desired. But because of really the revolution in DNA synthesis, it's possible to test many, many different designs. So once we have these designed genes, these genes encoding the designed proteins, we generally transform them into E. coli or transform them into yeast, where we can make the proteins and assay whether they have the desired binding activity. So if we design them to bind to the cancer biomarker, we see whether they actually bind, and we can do that either do that by in the case if we display these designed proteins on the yeast surface, we can do it by flow cytometry if we have a fluorescent a fluorescent version of the biomarker, or if we make an E. coli, we can test by surface plasmon resonance. So this is basically the workflow for most of what I'm going to be describing. So how do we actually do the calculations? Well, we start off with the structure of the target protein, and that's this surface view here. And the first step is to identify ways in which amino acid side chains, disembodied amino acid side chains, can make very favorable interactions with the surface. So this is essentially just taking disembodied amino acid side chains, docking them against the surface, sort of like identifying handholds and footholds, ways of really grabbing onto this surface. And the next step is to try and build a protein scaffold that will be able to support as many of these high affinity interactions as possible. So it's sort of like connecting the dots. We sort of have these disembodied hands and feet holding on, and now we need to build a body that holds them all in place so that we can make those interactions simultaneously and bind the target. So I'm going to illustrate this with the design of binders to the H1N1, the Spanish flu hemagglutinin. That's shown here. And in this picture, you see the structures of various antibodies that are known to bind the hemagglutinin, including one down here. We were very interested in this site because it's very highly conserved in influenza viruses, in part because it's right near the fusion peptide, which is critical for cell fusion, for entry of the virus. So if you blow up that site, this is now that in the stem region of the virus, there's this nice shallow pocket here. And we used the approach I just described. We identified placements for various types of side chains into that groove. So the groove shown in gray here, and we're placing these side chains in green and yellow into the groove. And then we can work backwards to see where the protein backbone would end up if we grow back from the tips of these side chains. You can see there's a number of different possibilities. That's what's in pink and blue here. So then we look for small proteins which can, on which these interactions can be, these residues can be placed simultaneously, and that's shown here. So this is the hemagglutinin surface again, and there are side chains that are docked into the surface here. Those are the ones that we identified, those interactions we wanted to make. And you can see it's a little hard to see, but this ribbon thing here is a small helical scaffold that positions all those side chains in the right places so they can make those interactions. And this is just another view from the other side. Here's an arginine poking into the groove. So as I said, our methods aren't perfect. We had to make a large number of different designs in order to find two that worked. You can get an idea of the scale by the fact this one's called HB80. Here it is, you see these side chains sticking into the hemagglutinin surface, which is shown in yellow, and the backbone of the scaffold is shown here. And here's another one called HB36, where we have a slightly different set of side chains poking into this groove, and here's the backbone here. I'm showing you these two because they're the ones which we actually find that bind the virus. They don't bind, so you could ask, well, why didn't the other ones work? And the way we are trying to get at that question is to use next generation sequencing. We made a library for each of those two proteins where we mutated every residue to every other residue. And then we use next generation sequencing to identify the frequency of each of those variants in the library before and after we selected for binding. And what this plot shows, this is sort of position in the protein versus amino acid type. And this, in places in red, which I think is a little bit hard to see, but there are some red, are places where an amino acid other than that which was present in the design actually was enriched in the selected sequences. So most of the places are green. That means that the design, that what the computer came up with was better. But the red places are things where there was something better that was identified in the selection. And we can use that information now. I mean, it's a very good test of our computer program because if our calculation was perfect, then there wouldn't be any way to make it better. But there are ways to make it better. And I don't think I'll go through this in detail, but we were able to see what the sort of things were that were missing and improve the computer model in Rosetta so that we were capturing those kind of changes. And it turned out that what they were primarily, what we were missing was the fact that the hemagglutinin is negatively charged in this region, and you actually can get quite a bit higher binding affinity if you put positively charged residues even at some distance from the interface. Those were the things that were enriched in this screen. Okay, so then Aaron Chevalier, who's a graduate student in the group, made another library where he combined all of those changes which were, all those mutations which are individually an improvement over the starting design. And it's really quite exciting. He got proteins which bind, the best one binds with 0.6 nanomolar binding affinity to the virus. That's better than the neutralizing antibody which binds to this region. And we were quite excited when our collaborators solved the crystal structures of these two proteins I've been telling you about bound to the virus. So here's the viral hemagglutinin. The design is, what does it say? In purple, that was where we thought it was going to bind. And then in red is the crystal structure. So it really binds. The crystal structure is almost exactly like the design model. You can sort of see that up here with this blow up. The side chains are really all in the right places. That was true for this design as well. So we only got 2 out of about 80 that we tested that had the desired activity, but those do bind in exactly the way that they were predicted to. So as you know, there are many different strains of influenza. There's group 1, which includes H1, H2, and H5. And these small design proteins bind to all group 1, essentially all group 1 strains, often even ones that aren't bound by the neutralizing antibody. And they block the conformational change that's required for the virus to enter cells. And not surprisingly, given that, they actually protect against H1N1 infection in cell culture. So this is now, the UW has licensed these to Johnson & Johnson, and they actually have a quite active program in developing neutralizing antibodies to treat the flu. And so we're very excited to see what happens, how far these small design proteins can go. And you can see the reason that they're excited about them. They're very small. They're under 50 amino acids. So you can make them in much larger quantities and more cheaply than you can make antibodies. And so this sort of led to the title of my talk in a sense. There's nothing specific about influenza or the flu or the hemagglutinin here. We just needed a target protein. And so in principle, you should be able to use this method to design binders to any any desired surface patch on any target of known structure, and maybe this is a route to new drugs. And as you know, efforts with small molecules have, it's been, despite a huge investment by big pharma and by NIH, there really aren't that many new small molecule compounds coming through the clinic. And one of the obvious problems is that small molecules just aren't going to have the specificity or affinity to tightly bind to one target and not bind to any others. Whereas you can imagine these type of small proteins might instead, which should in general have higher specificity and affinity than a small molecule. So that's sort of first example of designing a protein to bind to a pathogen protein. And if you have ideas about things, these things, I should say, we're also collaborating with Paul Jaeger's group here to use these small proteins as diagnostics, because again, for the same reason they might have advantages for therapeutics, they could be useful potentially as diagnostics because you can make them much more cheaply. So what about vaccines? With a vaccine, the idea is sort of the opposite. Rather than trying to bind a pathogen protein, you're trying to mimic epitopes on its surface, in particular those which are recognized by neutralizing antibodies. So there's work done with Bill Schief, who used to be here, but now he's moved to Scripps, and a student, Bruno Correa. So we did a lot of work on HIV vaccine design, but what I'm going to tell you about instead is on RSV. And the basic idea is illustrated here. Here's the structure of a neutralizing antibody bound to the RSV surface protein, the F protein. And you can see there's the actual part that's recognized by the neutralizing antibody is very small. It's just this little helical hairpin. So you don't really need to make, if you're trying to make a vaccine, you don't need to make this whole protein in principle. Maybe you could make just something minimalist thing that would display this helical hairpin. And so Bruno set out to do that, and the way he did it was to take that epitope, freeze it, and then try and build a protein around it. And that's what's shown in this ribbon diagram. He's searching for a good way, a good scaffold, trying to build a good scaffold that will hold this epitope in place. And as I showed you in those first two movies, after the initial search, there's a second stage where you try and add all the atoms in and design it to try and get something that would be really stable, at least on the computer. So Bruno then made these proteins, and it's really quite remarkable. The peptide by itself binds the neutralizing antibody with 200 nanomolar affinity, but it binds his designed protein, which is shown here. Again, this is a case where the crystal structure of the designed protein matches almost perfectly the design model. That's the overlay of the red and the blue. And the binding constant of this protein for the antibody is 30 picomolar. It's incredibly strong because this piece is held in place. And very exciting preliminary results suggest that this protein does elicit neutralizing responses to RSV in animals, but that's preliminary. So this basic technique of building, of scaffolding a peptide motif can be applied to many types of interactions that aren't necessarily between antibodies and antigens. And I'm going to give you three examples. One is the Epstein-Barr virus binding to BIM3. The second is MDM2 bound to P53. And the third is an interaction that's important in chromatin repression by methylation. And in all three cases, the question was, could we design proteins which would outcompete these interactions? Either this is a pathogen-host interaction or just a interaction between host proteins. And this is, again, something which is hard to do with small molecules. We're trying to compete out naturally occurring protein interactions. So Epstein-Barr virus, this protein BHRF, is a cancer promoter, and that's because BIM, the naturally endogenous protein, is a pro-apoptotic protein. It binds BCL2 and activates. That leads to apoptosis. So this viral protein binds to BIM and prevents it from promoting apoptosis. And so virus-infected cells are then more susceptible to transformation. So the goal was to block this interaction by designing high-affinity binders to BHRF1. I think I'll skip that. So we started with the interaction that was known between this BIM and BHRF1. And we again sought to build a scaffold. This is work by Eric Bracco around that protein. So we built a protein that presents the BIM binding, the peptide that binds BHRF in an optimal way. And this time we already knew that we were missing electrostatic interactions, so we put that into the model, and that actually yielded increased affinity. And the net result is shown down here, whereas BHRF binds the BIM protein with 24 nanomolar affinity, the design protein binds BHRF with 1 nanomolar affinity. So if we could express or somehow get this designed protein inside the cell, it should disrupt the BHRF BIM interaction. And the nice thing is that after optimizing specificity, it really binds. You don't really want to be binding all the BCL2 family members. This design protein binds BHRF1 much more tightly than it binds the other BCL2 family members that Eric tested. So we're excited about collaborating with Pat Statennews here on delivering this protein into cells which Epstein-Barr virus infected cells, to see if this will actually promote apoptosis by freeing up the BIM. And so those are just starting to undergo. Pat's been doing a lot of work on delivery of proteins into the cytoplasm. So the other two examples I wanted to tell you about is this EED-EZH2 interaction is involved in chromatin silencing. And you're probably familiar with MDM2 binding P53 and targeting it for degradation. And again, one of the reasons why design proteins are interesting for these, because these things are going on inside cells, there's no way of targeting these interactions with antibodies. So we just have preliminary results. There's a graduate student in the lab, James Moody, who's designed a protein which binds to EED. This is again, the protein that's a chromatin silencer, and he already has variants which bind considerably more tightly than this. And on the MDM4 front, he's actually trying to make proteins that will specifically bind MDM4 and not MDM2. He's got better affinity than the starting peptide. So this sort of illustrates how you can build off naturally occurring interactions to try and create inhibitors. Now, there's an obvious problem, which all of you has probably occurred to all of you, which is these designed proteins could be very immunogenic. And something which I'm very interested in doing, I think that I'm trying to convince someone in my group to do this now, is to see if we can make design proteins to be invisible to the immune system, or at least less immunogenic, by maximizing the content of peptide sequences found in the human genome and minimizing the content of MHC binding motifs. So because I think a lot of the potential of work in this area will depend on being able to design things that are not very don't elicit strong immune responses. Now, on the other hand, we're also, in the case of vaccines, we want to actually enhance their immunogenicity. So what we are thinking about is trying to design sort of a multivalent display platform that would have some adjuvant-like properties in addition to presenting the epitope. And I've been talking to a lot of people here about what TLRs and so forth might be good targets. So I'll sort of move on to this topic next. So if we look in nature, we see all types of different what you might call materials, like wires and spheres. These are containers and so forth. And so we've been making an effort, sort of spearheaded by Neil King, a postdoc in the group, see if we can sort of design self-assembling assemblies with the idea of making new materials, but specifically in the context of sort of this lecture for drug delivery and for vaccine presentation. Now, what we'd like to ultimately do is be able to build these out of completely synthetic parts. And towards that end, two postdocs in the group, Nobu and Rie, have been designing, sort of trying to understand the basic rules of protein folding and using these to make completely new proteins. This is just 4 proteins they really designed from scratch. They're not similar in sequence to any known protein. And the NMR structures have been solved. So these are the actual structures of their computed designs. So they made them in the lab, sent them to an NMR spectroscopist, and they're almost identical. So we can start designing building blocks, sort of like the bricks for the future, materials or other things we're trying to make from scratch. And the advantage of these is they didn't really evolve to do anything, so they're really very stable proteins, and they're now ready to be modified for a whole variety of purposes. But we aren't quite there to building symmetric assemblies out of them, so for now we're using naturally occurring ones. And the basic scheme is we've developed methods for placing cyclic oligomers into orientations where they would form the regular polyhedra. For example, these are 8 trimers placed to form an octahedron. And once we've done that, we can design these interfaces to stabilize the octahedron. And we've used this now to design synthetic octamers, and this is a tetrahedron. And I'm just going to show you pictures of those now. These are really neat. So these are the octahedrons. So we just make the protein in the cell. We get it out of the cell and then put it on an EM grid. And you can see these things are forming little cubes here. You can almost imagine rolling them. They're like little dice. It's hard to put numbers on the edges, though. And then here are just some projections of these cubes we've been making. And here's what the design model looks. But we have better than that. Here's the design. Here's what we were trying to make, this designed cube, and looking down the different axes of an octahedron, the 4-fold axis, the 2-fold axis, and the 3-fold axis. So we can really start thinking about designing assemblies with very high resolution. It's true even at the side chain level. And we have another one Neil's made now, which is a tetrahedron. So the application of this to the vaccine problem is sort of obvious. Now there's 24 copies of each subunit in this cube. So if we imagine hanging off, for example, that RSV immunogen, we'd have a highly multivalent presentation that would look to the immune system more like a virus. And if we add in binders to TLRs or the inflammasome, talking to Kelly Smith about that, then you could imagine having a very potent vaccine. More generally, you could think about using these things. We can use the same technique to create unbounded lattices, and you could imagine making new materials where you'd really be able to control where every atom was by, instead of making a closed polyhedron, say an open 2- or 3-dimensional lattice. Okay, so sort of in closing, I wanted to say that something about protein folding game players playing FoldIt have really been doing amazing things in the last 6 months. So the way that FoldIt started was a number of years ago. For the calculations I've been describing, we really didn't have enough computer power, so we started developed something called Rosetta at Home, which is a distributed computing version where you can download it on your computers, and it just runs in the background, and we get the results back. And we have something like 400,000 participants now. But when you run it, you get a screensaver shows up, and you see the course of the calculation. So people started writing back and saying, the protein's not folding up right. I've been watching these for a long time and getting a feel for it, but I can see it's doing the wrong thing. Can't you give us some way of guiding it? And so that's where the idea of FoldIt came up. We teamed up with Zoran Popovic's group in computer science, sort of make an interactive version which sort of does the same basic energy calculations, but instead of the search being random, now a person can guide it. If the person wants the computer to do some work, the person can push various buttons and tell the computer what to do, or the person can actually go in and move things around themselves. So there's 3 really quite amazing things that people have done in the last 3 months, or maybe 6 months now. One is that we've been developing methods for solving protein structure I didn't really talk about today, and we had one particular protein we were stuck on. We had a data set that a crystallographer had collected, but no one had been able to solve the crystal structure. And so we set it up as a challenge to FoldIt players, and somewhat miraculously, they were able to solve the structure. At which point I learned you shouldn't ever publish papers that have the word games, HIV, and in them at the same time, because it led to a media storm, which you may have seen. the second thing, which is probably was prompted by the fact that clear that people were doing non-trivial things, and so we wanted to know what those were. So I wrote to the top FoldIt players and asked them what they were doing, and I got back these 5-page detailed emails of all the super complicated and incredibly neat things they were doing. And it was so, we had thought, well, we could put them back into the Rosetta program and make automated prediction work so much better, but it was really complicated to see how to do that. So we decided instead to let the FoldIt players figure it out. And so we gave them the ability to write simple scripts and algorithms, and to our surprise, these really took off in the population. And what they ended up coming up with was eerily similar to the best thing we'd ever come up with in the group, but not published. And they actually improved on it to some extent. So the game players have actually been developing improved algorithms. And what I want to talk about today, or tell you about briefly, is how FoldIt players have actually been designing proteins. Because in a way, you might expect this to be the place where human creativity could have the biggest impact, because whenever you design something or make something, we think of that as more intrinsically a human process than a computer process.
A really good FoldIt player,
yeah,
I think.
Sorry. The reason I'm showing you this video is people always ask me who FoldIt players are, and I really can't do better than show this clip.
A really good FoldIt player, I think, needs a number of skills. Problem solving is obviously one of them. I think a slightly addictive personality, a little bit of stubbornness thrown in, and a little bit of luck on top. You don't necessarily need to have a scientific background to play, you just need to know what works and what doesn't. And you can learn that within the game itself.
I'm an administrative worker in the rehab team. I'm just answering telephones, working on bespoke computer programs, interacting with staff. It's very repetitive. When I go home, I become a different person. I just like to measure myself against something, and it's given me something that my everyday life hasn't given me. It's to just use abilities I didn't know I had.
For me, it's a guilty pleasure, and yet here I am involved in something that has real relevance in the scientific world. It makes you feel very proud of what you do, which is essentially a little hobby.
Right. Well, so that was a much more eloquent description of who FoldIt players are. Those are two of the world's top FoldIt players, by the way. So, all right, so we've also been doing work I didn't describe on designing catalysts for chemical reactions where there aren't currently catalysts. And so this is, for example, a catalyst we designed a couple years ago that forms 2 carbon-carbon bonds between these 2 substrates. And here's the enzyme, a schematic. It binds them together in such an orientation that promotes catalysis, promotes the reaction. So here's a model of that. Here's the enzyme is shown in yellow, and the 2 substrates are in purple. And you can see how it brings them together. And when we made this protein, this is work by Justin Siegel and Alex Angelini, we found that it was quite active, which was neat because there aren't naturally occurring enzymes for this reaction. On the other hand, it was not a very good catalyst, and we were kind of stumped on how to make it better. So Chris Iben, a undergraduate who had been playing a lot with FoldIt in the lab, thought that, and Justin thought this would be a good challenge for FoldIt players. So when you look at these 2 substrates bound in the enzyme active site, you see the active site is really totally open on one side. There's nothing here. And so you could imagine if you're really rebuilding this thing, you might want to build some brand new part of the protein that would come up here and make a better active site. But how to do that, that's not something you can ask a computer to do. A computer is not smart enough to figure out how you might do that. So instead you ask protein folding game players. And they came up with this rather crazy thing. They added 15 amino acids to the sequence and remodeled about 25 residues total to build this big helical hairpin here that you can see does exactly what solves the problem we wanted to, at least on the computer, in that it sort of cradles these 2 substrates. And the really cool thing is when Chris made this protein, I never thought this was going to work. It actually turns out to be about 20-fold higher activity than the design, which is quite a lot. And this is definitely now the best catalyst of enzyme or non-enzyme known for this reaction. So that was pretty neat. And then Barry Stoddard's group at the Hutch was able to solve the structure. And this was really even more, almost as maybe even more remarkable, which is that the structure of the actual structure, which is shown in brown, is almost identical, or is very similar to what they had designed here. So, and in retrospect, it really wasn't so crazy. I mean, my enzymologist colleague Don Hilbert says this is totally ridiculous. It's like you've built this whole thumb on top of the active site. But the fact that it's a helical hairpin and can buttress itself, I think, is something now we're trying to apply within the lab to get stronger interactions to other proteins. So this was to bind an enzyme, but we're now, one of the things I mentioned was H3, group 2 influenza, and we're now challenging FoldIt players to help us bind proteins that will bind to group 2 influenza viruses and hopefully may one day have therapeutic potential. And that I think will be pretty neat when a game player. We've already overcome the hurdle of getting players F indexed in Medline. When a journal complains, we just show them that players F is already well represented in Medline. But if they can actually develop a therapeutic or something, that will be really change the relationships between science and society. All right, so I've been really lucky to have fantastic coworkers. So the inhibitor work was done by Cyril Fleischman and Tim Whitehead, who are both professors at other institutions. They've left in the last year. Aaron is a student in the lab who did the deep sequencing work. Eva worked on the methods development. Yifan improved the computational model. Eric did the work on the Epstein-Barr virus protein inhibitor, and James did the work on the other two protein interactions. On the materials side, this has really been inspired. Neil King really had the idea to do this when he came to the group. He's been working with Will Scheffler on the methods. Nobu and Rie have been trying to make ideals, or the Platonic ideal versions of proteins. I showed you some of them. And the structures were solved in Todd Yates and Tamir Gonin's lab. FoldIt is a collaboration with Zoran Popovic's group, and Seth Cooper has really done a lot of the key development work. Firas Khatib has really been involved in all of the aspects of FoldIt I described, and setting up the puzzles and looking at what people are doing. And Justin and Chris did the work on the design. Bruno did the work on the vaccine development. That was a collaboration with Bill Schief. And again, I mentioned the Institute for Protein Design that we're starting up here. And if you have ideas about how to apply these methods to diagnostics or therapeutics or anything, please let me know, and I'll stop there.
Well, David, thank you for a wonderful talk. Remember, computer games, it's not just fooling around. This is fantastic. But I'm sure there's some questions.
Yeah.
Does the approach work for oligonucleotide aptamers, and if you tried that?
Let's see. We have not tried. We've done some. We've used our prediction methodology to predict RNA structures, but we haven't done any explicit RNA design work. One of the issues is that RNA folding is considerably earlier on, and so while one can design a sequence and have a reasonable probability that will fold up if it's a protein, it's a little bit harder with RNA. The other thing is that proteins have a real advantage in that you have 20 different types of amino acids. So your opportunities for catalysis or for binding are quite a bit richer than RNA. So as long as you're designing things, you might as well choose a toolkit with 20 different compartments rather than one with 4. Yeah?
Unfolding and refolding experiments with your engineered proteins, what percentage properly fold?
Well, yeah, it's a good question. So remember, in any of these experiments, only a minority of them have the desired activity. So something that's possible for the ones that don't bind is they're simply not folding properly. And so the ones that come through that I've described all fold and refold reversibly. There's no problem at all. But it very well could be that the ones that we're losing have folding problems. It's hard to convince crystallographers to study the ones that don't work. Even people in my lab, it's hard to convince. So we don't really know as much as we'd like about why the ones that don't work. Yeah?
Is the primary bottleneck in this work the computational complexity or the completeness of the model, or some combination? If you had infinite computing power.
Yeah. I would say that Actually, for a lot of the work we'd like to do, the bottleneck is more on the biology side. If we want to design an inhibitor for want to design a protein that binds to a TLR, for example, it's getting that protein because it's very hard to make toll-like receptors. So a lot of the slow part is just getting the protein, the target. Once we have the target protein, then I would say the bottleneck is, it's sort of spread out. I mean, you can only, one of the things that people spend a lot of time on in my group is you can only currently test on the order of 50 or 100 designs, and the computer will spit out tens of thousands, so you have to filter them in various ways. But there's higher throughput DNA synthesis coming online, just in the same way that DNA sequencing took off. So I think in the next, soon we'll be able to make many more designs, and so that will, so it's not totally clear what the bottleneck is, but the most immediate one I can tell you is just getting the target protein that we can do the binding experiments on is certainly a limiting thing. Yeah.
Is there a size limitation such that if the proteins get too large, either the models don't work or the computer isn't available to sort of do that kind of modeling?
No, there's not really any size limitation. I mean, hemagglutinin is big, so the target can be any size. And in general, for the design protein, you want it to be smaller rather than bigger because you're thinking about manufacturing it in large quantities down the road. So there isn't really a size limitation. And for example, these symmetric assemblies get to be quite big. There certainly would be a size limitation if you're trying to predict the structure. It's just the methods work worse and worse as the protein gets bigger. But for design, that doesn't really hold so much. Yeah.
Is getting these proteins into cells a problem?
Yeah, well, that's a key question. So I mentioned that we're collaborating with Pat Staten. He has these polymer-based delivery things. But the whole delivery question is really important. I mean, the hemagglutinin, obviously, that's if you want to block the flu, you don't have to get the proteins into cells. But as you probably know, there's been a variety of efforts on how to get proteins into cells. You could imagine doing it with viral vectors. You could imagine doing it with polymers, and so that's still one of the things that would need to be worked out if any of these are going to be useful. Any of the intracellular ones would be useful. Yeah.
For the oligomers, how efficient is the assembly of those with the designs you have?
Yeah, it's right for the ones that work. Again, we test a number to find a few that work, but the ones that work assemble very, very fast. So that's 100% efficient.
And then if you functionalize them, does that affect the...
Yeah, so we're just starting to do that, and so we don't really have the answer yet. I think some of them will... The problem with the cube is you've got all these termini that are really close together, so there may be steric collisions. But that's obviously for the vaccine application, that's an important question. And I imagine that in some cases it'll be easy to put anything on, in other cases it'll be difficult. Yeah.
So, I think you won 3 out of the first 4 structure prediction competitions. The CASPs, when they first came out, your lab did.
Yeah.
If you could have employed the FoldIt players, would you have won?
Well, let's see. So, in fact, FoldIt players are very excited because the 10th one, CASP 10, is about to start in May, and they're getting psyched up to do well this year. They're going to be a completely independent team. So the answer to that will become clear. Yeah.